All right. So my review is on current ecocentric models of color perception. It's a little technical, so let's get right into it. First, we're going to do an introduction to the retina, because even though I know retinal physiology pretty well by this point, I'm pretty sure you guys don't. So I want to get you a good ground. Then we'll talk about ecocentric versus anthropocentric ways that we answer biological questions. We'll look into physiology and behavior and look at these as different questions we can ask to determine what animals perceive. And then we'll just get into some different models of how different animals can see things. So starting off with humans, then going into a nice, easy mice model, which is very similar, then into crustaceans and cephalopods. And then we'll have our closing remarks and acknowledgments. So a quick introduction to the retina. The retina is made up of, the human retina is made up of many layers of different neurons that process the light a little bit before it gets sent back to the brain where it's further processed. The first layer of cells to interact with light is the photoreceptor layer, unsurprisingly. They transmit the light signal to the bipolar cell layer, which then is mediated by horizontal cells, and they kind of do a little bit of processing. Next, amacrine cells also do more communication before the signal is sent to retinal ganglion cells and then back to the brain. All right, so big key takeaways: light hits the photoreceptors, multiple layers of neurons mess with the signal before it goes back. And so what we're going to talk about in this lit review, let's get into this, is anthropocentric versus ecocentric models of color vision. Ecocentric questions are things such as what chemicals do they use, or anthropocentric questions focus more on human behavior and human anatomy. And these questions aren't necessarily mutually exclusive. If we were to ask a question about for example, the sponge chemical usage, we can then turn around and say, can we use this signal, or can we use this chemical to treat cancer in human beings? How else can we use this? We also look at questions from a bottom-up or a top-down perspective. We can study the physiology and the nitty-gritty of how far apart are the photoreceptors and how does this affect how small of an object can they discern. Or we can look at their behavior, and we can ask, if we were to use a UV filter, would the mating display of a jumping spider be as effective? And so this is where anthropocentrism can come and become a bit of an issue. Because when we look at physiology, we know so, not to disparage research into human physiology, however, we know so much that when we look at animals, we often assume human physiology. And when we look at behavioral studies, this can often confuse us. So after studying human physiology can be a problem when it comes to trying to understand how animals see. So now that I said that, let's start off with the standard model, which you'll get taught how color vision works in a neuroscience class. The standard model is the human model, and it's based on opponent processing. Different photoreceptors have different peak sensitivities. So for example, a blue photoreceptor is maximally sensitive to light of a wavelength of 420 nanometers, which we interpret as blue. Whereas green and red are maximally sensitive to green and red respectively, but they can receive light from a large swath of the color spectrum. So as a way to get an efficient and clear signal out of that, humans use opponent processing. So if this was a retinal ganglion cell, it responds to both green and red light. However, it gets a positive input from a green photoreceptor, and using horizontal cells that I mentioned earlier, it inhibits the signal from a red photoreceptor. This is why we can't imagine a reddish-green light or a bluish-yellow color, because these color systems are antagonistic to each other. So the main takeaway of how the human system works, we've got 3 cones involved using an antagonistic system. There are rods, but rods do not play a role in our color perception. So let's get our feet wet with an easy mismatch between behavior and physiology. If I were to peer into your pupil and try to take a picture of the back of your eye and look at what photoreceptors were on the back and lay it out, we could look at the different sizes between photoreceptors and see that there's a different distribution of photoreceptors across your retina. If we were to look at the cones in mice, we could see that in the ventral retina, it is composed solely of ultraviolet cones, whereas the dorsal retina is composed of a mixture of ultraviolet and green cones. So based on what I said earlier, you would expect that in the ventral retina they'd be colorblind, because as I said before, you need a contrast between two different chromatic sensitivities to determine color. So but when they do behavioral experiments, what they find is that these guys still use their ventral retina to determine territory markings using ultraviolet reflectance from urine trails. So how do they do this? Well, the dorsal retina works exactly as you expect. The cones, both colored cones, work as an antagonistic system. However, in the ventral retina, what happens is the rods actually pitch into the color-sensitive pathway, and they use their rods to create this antagonistic color system. So that's an easy example of how assuming human physiology that only cones contribute can be a mismatch and limit what actually occurs in biology. This is also used by other mammals, such as aquatic mammals that only have one type of cone, and they use their rods primarily for their color vision. Now let's look at mantis shrimp. These guys, you may have heard there's a Discovery Channel clip out that talks about how they have amazing color vision because they have 12 photoreceptors. And if they use this opponent processing mechanism with these many photoreceptors that cover all the way from ultraviolet light, just barely getting into like infrared, you'd expect they'd have incredible color vision. So when they did a behavioral experiment where they presented them with colored targets of different wavelengths, they expected that they'd be able to tell the difference between 5 nanometers. So if this projector is right, there should be a 5 nanometer difference between this target and this target. However, when they did the experiment, what they found was the mantis shrimp were only able to discriminate between like 12 and 25 nanometers. They couldn't see as well as even we can. So why can't they see color as well if they have so many photoreceptors? Turns out they use a different mechanism. If you were to shine a light of this color up there to their retina, and we were to look at how these cells would respond, they probably wouldn't respond much because their maximum sensitivity is UV. Whereas these guys wouldn't respond much because it's red. Whereas these guys would respond a lot because the sensitivity is of their preferred stimulus. So we'd expect to see something like that, and we'd guess that that's where the color is. Likewise, if we were to do it with ultraviolet, you'd guess over there, and that's kind of what color you'd expect. The way this works is instead of using an antagonistic pathway, they just guess based on a general summation of firing patterns. This doesn't give them as high an acuity as we would get with this many photoreceptors, but what it does give them is efficiency. A mantis shrimp is a predator that punches really fast, essentially. And each of their punches that they do takes place in 1/50th of a human eye blink. So they really need to live in a fast perception world. And so the advantages that they gain from having this fast perception outweigh the cost of not being able to see 5 nanometers of wavelength difference. All right, and so now for my favorite guys, these octopuses, cephalopods. These guys have, if you look at the top picture, can you see the octopus in the first one? What about the second one? Can you see it in the last one? These guys have incredible camouflage, and the squid there is communicating to different squids on either side of its body. They use color a lot. But they only have one photoreceptor. This should strike you as odd, because in all of these so far, they've at least compared something somehow. So how are they doing a comparison with only one photoreceptor type? Well, their eyes are remarkably similar to ours. Very advanced. They have a cornea, retina, lens, vitreous humor. The structure is very similar. But the one thing that's different is their pupil shape. The one thing that's different besides their photoreceptors is their pupil shape. Their pupil shape works a lot like a triangular prism. You know how the triangular prism separates light and splits it out into its rainbow wavelengths? With these guys, what happens is their pupil shape causes light to focus at different focal points based on its wavelength. So a light with a short wavelength will focus in front of a long wavelength. So what happens here is when the ciliary muscles are relaxed, blue light or an ultraviolet light would focus on the retina, whereas a red light would focus behind it and be blurry. Then if you contract the ciliary muscles, round up the lens, it would bring the red light to focus, but as it brings the red light into focus, the blue would blur. Because of this difference between colors, they're able to sense which color or which wavelengths of light is in focus. And by doing this alternating focus mechanism, they can determine what color a light is. This is really interesting because in a lot of laboratory experiments over the years, they've concluded that octopus are actually colorblind. However, as you've seen in the behavior, that can't be the case. So there's been a lot of why is this working? Over the past 60, this model, though it hasn't been tested on animals yet, it's been done on MATLAB, it does align with the past 60 years worth of cephalopod behavioral data. For example, one reason that octopuses may seem colorblind in a lab is if they're using fluorescent lighting. Fluorescent lighting is flickering out different wavelengths of light constantly. So if your color perception is based on comparison, but you're getting a flickering input, you wouldn't be able to make a clear comparison as you would as if you were getting a pure sunlight constant illumination. So we're going to make some closing remarks. Human models are very useful for informing our animal models and very useful starting points. We wouldn't get nearly as far in studying mouse if we had to assume everything, assume nothing from the beginning. But behavioral studies can clarify where we're missing things and what we're forgetting to understand. There's no such thing as one model of color vision. There's no such thing as one way that it works. Like with the rest of biology, there's no one way to do things. And there's so much that we don't know. Yeah. So I wanted to give a huge thanks to Dr. Mukba for helping me so much all these years and for the help of this lit review, taking me on last minute. Thanks to my peer reviewer and for Dr. Ford. And yeah, if anyone has questions, take them now, or if you have questions later. I had to cut a lot of stuff to get this in here, so I cut probably like half of my lit review. So if you have questions of other models, I can also answer that.
So most mammals do not have red cones, correct?
Correct.
And so they do not see red, and they don't have this processing system you're talking about that the octopus has
Yes.
to figure it out by a focus difference, right?
Yeah.
So why octopuses? Why not deer and squirrels?
So why did I focus on cephalopods instead of deer?
No, why would it work? Why would the octopus have this and not other mammals? You think mammals would be important in mammals
Yeah.
as much as in an octopus?
There's a lot of, I don't know why the color red would be useful to most mammals, because my understanding is color vision evolved in primates primarily to tell about ripening fruits,
Yeah.
whereas I don't know what behavioral specifics that red would be useful. However, if you look with the cephalopods, it is really useful to see red because red is their major threatening color, so it is behaviorally useful to them.
Yeah.
I could also say that the red photoreceptor is a mutation of the green photoreceptor. And so they actually have done a study that I find really interesting where they put a red photoreceptor in a mouse. They added that to the genome of a mouse, and the mouse did develop trichromatic color vision from the introduction of that photoreceptor. So they have the rest of the hardware already embedded to see trichromatic, but they don't have the photoreceptor.
One thing that has always been interesting to me is red is a danger in nature.
Yeah.
So other mammals are at a disadvantage to humans and primates.
Yes.
They can't notice poisonous plants and poisonous animals,
Mm-hmm.
which often are colored red.
That's very true, yeah.
So it's a mystery.
Mm-hmm.
Are there so in humans, the red and green cones are carried on the X chromosome,
Mhm.
which is why colorblindness is obviously more prevalent in men, especially in red-green. My question is that also true as far as certain cones being carried on sex chromosomes in other animals?
Mhm.
Because obviously their sex chromosomes are different than ours.
Yeah.
I mean, you think of a bird where the
That's a
homozygous
very
is
good question.
the male. So
Yeah.
now I'm curious, because I know birds are tetrachromats.
The only piece I put about birds, the only piece I looked into about birds is their whale droplet filters, so I did not look into if a bird can be colorblind. That's a very interesting question, though.
I don't know how one would go about finding
I
their
could-
study
I could-
population.
I could talk to Esteban, the guy I'll be working with at Purdue, and see if he knows anything. That's a good question.